Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for the opportunity given by the 6th EcoBTD Committee. Today, I will be present our research in a title, Acute Toxicity Test of Moringa oleifera seed oil in rats, potential use as an oral cavity treatment. Background. Moringa plant or Moringa oleifera is a tropical plant that is widely consumed by Indonesian people because it is rich in nutrients. Moringa plants can be used as whole starting from the roots, leaves, fruits, flowers, and seeds. Moringa seed contain flavonoid compounds, tannins, saponin that function as antibacterial and anti-inflammatory, which can be used as medicinal ingredients, especially in the oral cavity. Objective. This study was to evaluate the toxicity of Moringa oleifera oil with parameters of hepatotoxic domains based on liver enzyme in rat as a treatment oral cavity. Method. This type of research is an experimental laboratory with post-test only control group design. The SGOT and SGPT examination were used to measure the level of toxicity. Toxicity test. This study, we use a white trace, Rhetus noverdicus Vistar strain for experimental animals. Indicator, liver's disorder indicator through SGOT and SGPT examination, and the result is a 625 milligram per kilogram and 1,250 milligram per kilogram doesn't cause liver impairment. Safe dose 2,500 milligram per kilogram and 5,000 milligram per kilogram cause liver impairment indicated by significantly different SGPT and SGOT value. Toxicity test. Materials, 20 white rats, Ratur noverdicus, consisting of 10 males and 10 females, adapted for 7 days in lab environment. Moringa seed oil was obtained from South Sulawesi, while chemicals were purchased from registered chemical distribution. All diagnostic kits were purchased from Human World Diagnostic Germany through an official distributor. Blood sample collection. Blood sample of rats were taken through the lateral veins after anesthetized with either through inhalation. Blood samples were withdrawn using 3 cc syringe and collected in a container tube containing anticoagulant. Plasma separation. The blood was centrifuged and rotated for 20 minutes at a speed of 2,500 RPM. The blood serum was removed with a micropipette and transferred to the Eppendorf tube and stored in the refrigerator at 4 degrees Celsius until further analysis. Moringa oil preparation. Moringa seed oil was dissolved in corn oil, the solvent in a ratio of, and as for the calculation of those are moringa 625 milligram per kilogram was obtained from dilution 3.5 milliliter of moringa oil in corn oil to make a 20 milliliter oil mixture. Moringa 1,250 milligram per kilogram was obtained from dilution 5 milliliter of moringa oil in corn oil to make a 20 milliliter oil mixture. And moringa 2,500 milligram per kilogram was obtained from dilution 10 milliliter of moringa oil in corn oil to make a 20 milliliter oil mixture. And the moringa 5,000 milligram per kilogram was taken from stock solution without adding corn oil. Acute toxicity test. In this study, 20 rats were divided into 5 treatment groups, each consisting of male and female rats. To administer an accurate dose. Each rat was weighed and the dose calculated accordingly prior to Moringa seed oil oral administration. The 5 treatment groups are: the first group, healthy control only given standard food and drink, and the second group, given Moringa seed oil of dose at 625 milligram per kilogram. Third group, given Moringa seed oil at a dose of 1,250 milligram per kilogram, and the fourth group, given Moringa seed oil a dose of 2,500 milligram per kilogram. And the fifth group, given Moringa seed oil at dose of 5,000 milligram per kilogram. A good toxicity test. After that, the rats were observed for 7 days for clinical signs of toxicity, such as lethargy, diarrhea, and weight loss. After 7 days, the blood samples were collected to evaluate the SGOT and SGPT analysis. SGOT and SGPT analysis. Blood samples were sent to the Clinical Pharmacy Laboratory, Faculty of Pharmacy, Unhas, to analyze liver function tests, namely serum glutamic oxaloacetic transaminase, or SGOT, and serum glutamic pyruvic transaminase, or SGPT. Increased level of SGOT and SGPT will occur if there is an intracellular release for enzyme into the blood caused by necrosis of liver cell or acute damage. Or in other words, level of SGOT and SGPT will increase in the blood when there's damage to liver cells. Result. This is the table, the difference in the mean value of SGOT and SGPT. And then the shape of Moringa oleifera lamp is high dose was evaluated on the liver function of experimental animals by measuring SGOT and SGPT, and the result can be seen in this table. One-way ANOVA statistical test on the SGOT and SGPT value obtain a significance value of 0.020 and 0.000 respectively. P less than 0.05, which means that there is a significant difference between the treatment groups. Furthermore, a further reference test, post hoc with LSD test, was carried out to determine the difference between the two variables. In healthy control, when compared with a dose of 625 milligram per kilogram and a dose of 1,250 milligram per kilogram, the p-value greater than 0.05, which means that there is no significant difference or has the same effect. Mainly the healthy control, when compared with a dose of 2,500 milligram per kilogram and a dose of 5,000 milligram per kilogram, had a p-value less than 0.05. Which means that there is a significant difference or has a different effect. The result of the LCD for two different tests on SGPT show the healthy control were compared with a dose of 625 milligram per kilogram and a dose of 1,250 milligram per kilogram had a p-value greater than 0.05, which means that there was no significant difference or had the same effect. Mainly the healthy control were compared with a dose of 2,500 milligram and kilogram and a dose of 5,000 milligram per kilogram at p-value less than 0.05, which means that there is a significant difference or has a different effect. The table shows they use a dose of 645 milligram per kilogram to 1,250 milligram per kilogram. There is no difference in the value of SGOT and SGPT with healthy control. So that the use is a dose of no more than 1,250 milligram per kilogram is still in the safe zone, while the use of dose of 2,500 milligram per kilogram until 5,000 milligram per kilogram show a significant difference in the result with healthy control. So it can be said the liver damage or toxicity has occurred at that dose. Discussion benefit of Moringa. Moringa is source of nutrition. We know Indonesian people always consume the moringa because of nutrition. And moringa medical ingredients like flavonoid, saponin, and tannins, and they have effect for antimicrobial and anti-inflammatory. Liver damage. Toxicity test in this study, white rat, is experimental animal by using indicator of liver disorder through SGOT and SGPT examination. The liver is one of the toxic target organ. Liver remains is caused by unbalanced ions, fluids, or metabolic product. Liver demand can be assessed by increased activity of SGOT and SGPT in blood. SGPT can reach 20 to 100 times above the highest normal limit when liver necrosis occurs. Increase of SGOT and SGPT was caused by increased oxidative stress, so that the production of oxidant and free radical increase. The number of cellular antioxidants remains, so they are unable to fight free radicals or oxidant attacks. The membrane lipid peroxidation reaction by free radicals directly impact on cell membrane damage, fluidity, change, cross-linking, membrane structure, and function. If the condition occur continuously and extreme, it may lead to cell death. Result. Toxicity test show that the safe dose that can be used is no more 1,250 milligram per kilogram, while toxicity occurs at a dose of 2,500 to 5,000 milligram per kilogram. The result of statistical tests also show a significant difference between the control and the test group of 2,500 and 5,000 milligram per kilogram. Conclusion. Moringa oleifera is a safe use with a dose, if not more than 1,250 milligram per kilogram. Thank you very much.